Good evening, and thank you so very much to the organizers of the NASPGHAN Visits Last Began series of webinars. Tonight, I'm going to talk about the role of the microbiome, probiotics and prebiotics in health and allergic disease. And I hope I can stimulate some thought, generate discussion, and talk about where some of the exciting science is going in terms of understanding the origins of allergic and inflammatory disease. Again, thank you so very much for the organizers, for the invitation and the opportunity to present to you this evening. And in these webinars, it is our hope that we stimulate ongoing collaborations between our two organizations. So tonight's objectives are as follows. I'm going to hope to provide you data which describes how diet, environment, and the microbiota in infancy affect long-term health, and how the intestinal microbiota support gut barrier function and modulate the host immune response. In addition, I'll talk about factors that affect how microbes colonize the intestinal tract and how these microbes contribute to the development of a healthy immune system, and how this represents in particular an area where we potentially can intervene, not with pharmaceutical agents, but with actual dietary approaches, pre and probiotics, to actually influence genetically predetermined outcomes. As a card-carrying epidemiologist, I think it's important to understand the context of the diseases I'll be talking about tonight with respect to its epidemiology. So one of my objectives is to review the prevalence of allergies in the United States, as well as other autoimmune diseases, and the clinical surrogates of allergy early in life that may predict allergy later in life. And I'll provide some evidence regarding dietary supplements, in particular pre and probiotics, and this concept of symbiotics, or when you combine the two, and changes in the gastrointestinal microbiota that can influence disease outcomes. So the first section, and I've divided this talk into sections so that it compartmentalizes it into telling a story. The first part is the microbiome, immune development, and defining for you this concept of dysbiosis. It's important to recognize, and I'm biased as a pediatric gastroenterologist, that the GI tract really is the center of lots of things, and in particular, our immune health. And the intestinal microbes that colonize the GI tract, we're now realizing contain a wealth of information, and their interreaction with the host immune system within the gastrointestinal tract can influence outcomes within the GI tract and in other organ systems throughout the body. The gut microbiota helps support gut barrier function by helping enhance mucin production, decreasing permeability, and in terms of the immune response, actually generating IgA activity, balancing T helper cell subclasses, and importantly, enhancing the traffic cops, if you will, of the immune system, and balancing between the T helper 1 response, which when exaggerated results in autoimmune disease, and the T helper 2 response, which when exaggerated causes allergic disease. And what gut microbes do is interact with the T regulatory cells, which can shift from one way to another, the T helper response. It's also important to recognize that the microbiome is made up of the microbes, the microbiota, and their products, which actually have benefit as well. And it's a very tightly controlled community or ecosystem between good and bad bacteria. So potentially harmful bacteria, when there's an imbalance, can result in things like diarrhea, altered motility, and importantly, a decreased diversity in the microbes in the gastrointestinal tract can result in disease, such as allergy and autoimmune disease. On the other side, potentially helpful bacteria can inhibit things that you ingest, either through what you breathe or what you eat, and it stimulates immune functions and results in healthy immune development, as well as things like synthesizing vitamins and micronutrients. Now, it's important to recognize that for a long time, we felt that at birth, there was a sterile sort of gastrointestinal tract, if you will, and then how you were born and what you were fed, whether you were breast or bottle fed, that influenced the microbes that were in the gastrointestinal tract. And the most variability was early in life. in this elegant study published about 6 or 7 years ago, where they looked at fecal samples from birth through elderly, and they showed that the variability was most seen in children under the age of 3 in person-to-person evaluations. And it's that period under the 3 years of age where the gut microbiota is most susceptible to change, as well as response to environmental factors. So diet, or on the bad side, more exposure to antibiotics may influence health outcomes. And there are studies that have been published now over the last decade or so that have highlighted these things. 2 of them on this particular slide, the American Academy of Asthma, Allergy, and Immunology, infant gut microbes shape future health and allergy risk, or a paper published during that same year, newborn gut microbiome predicts later allergy and asthma. So there's been a lot of attention on the infant gut microbial composition and how it shapes the development of health or disease, and whether there's a role of genetics. An even more provocative study just published within this past year, a multi-center, multinational collaboration that has actually been characterizing the gut microbes from birth through older ages. And they actually identified that it may not necessarily be all too late after 3 years of age. And that there are studies that demonstrate even in preschool and primary school age that you could potentially influence the gut microflora. And in fact, in some of the studies that published in this paper just about a year ago, even teenagers. The take-home point here is that the microbiota may develop more slowly in some children than others, and children may provide beyond the age of 3, a potential opportunity to use microbiota-based interventions to either promote health or prevent dysbiosis and disease. Now, let's look at this in a schematic way, and that's shown on this particular slide, which shows maturation of the gut barrier function in the immune system. And what we've realized now is that initially there are some microbes in the gastrointestinal tract of infants, and I'm going to show you some very recent provocative data, at least what we used to think, and that it was initial colonization in the mode of delivery and the exposures either through the mother's vaginal tract to intestinal vaginal flora, as well as skin microflora that greatly influenced the microbes and the microbiota that the infant was initially colonized with. And there's a lot of data looking both at comparing either C-section or vaginal delivery, and I'm going to talk about this later in the presentation, and influence on health outcomes. And then in terms of the type of feeding that the child undergoes, whether they're breastfed or bottle fed or both, has a role in enhancing the diversity, where at least in microbes and as in people, diversity is good. And so increasing the diversity results in a healthier microflora. Well, just recently in the past year, some very exciting data taken from infants in utero demonstrates that in fact bacterial colonization begins even within the first trimester of pregnancy. But so between 18 to 20 weeks, you can actually have bacterial colonization of the gastrointestinal tract. And these studies published in Nature Medicine of last year, 2 different studies from different groups, demonstrated that there were a number of bacterial profiles identified, in particular, a specific microbe, M. luteus, that actually promotes immune regulation. So if you think about that, even in utero, foundation or basic microbes lining the gastrointestinal tract, influenced with the very immature and developing immune system within the gastrointestinal tract, and result in eventual healthy outcomes. This particular bacteria was actually shown to reduce pro-inflammatory chemokines, interferon gamma, by fetal memory T cells. So some really provocative data to demonstrate that even in utero, the development of the microbiome is starting to happen and its relationship to the host immune system. So if you think about it on this schematic slide shown here, each person, just like you have a unique fingerprint and you have a unique genetic makeup given to you by both mom and dad, each person develops a unique microbiome. And that's influenced by the diet that you have, whether you're breast or bottle fed, how you were delivered, whether you are preterm or term, and then your genetics, as well as the environment on which you were born, whether you were raised on a farm and exposed to lots of animals, or in a city in an urban environment, and whether you got antibiotics. And it's the combination of all of these that result in either a healthy immune system or that result in this concept we call dysbiosis, which results in disease. So infant exposures help define the intestinal microbiota. And to sort of show this in a different way, these circles represent different exposures to infants early on in life, where the green are those exposures that enhance a more diverse and rich microbiome and thereby a more healthy, robust immune system. And the red are bad, things that actually decrease diversity of the microbiome, C-section, antibiotics, premature hospital delivery, and result in a non-healthy or a dysbiotic microbiome. And it's that dysbiotic microbiome that results in the specific diseases that I'll talk a little bit more about later in this presentation: asthma, allergies, atopic dermatitis, or potentially could prevent them. So think about the body's microbiome as being rich microbial communities in each of the compartments within the body. There's data actually in the otolaryngologic literature looking at the microbiome in the ear and its relationship to the development of ear infections, or in people who are chronic contact wearers and a dysbiotic microbiome that's on the surface of their eye, giving them more predisposition to developing corneal ulcers. And so that gut microbiota working together with the host makes a superorganism. And it's important for you to understand that it's not just bacteria, it's fungi, it's viruses, it's parasites and protozoa, all of which make up these rich microbial communities interacting with the host to promote either health or disease. And so one of the concepts to take home tonight, in addition to understanding how early the microbiome actually starts developing, is one of dysbiosis. Dysbiosis is defined as the perturbation of the normal microbiome that results in a disruption of the relationship between the host and the normal microbes. And that disruption can then result in diseases such as IBD or other gastrointestinal disorders like IBS, irritable bowel syndrome, or even cancer. And I put that sort of pictorially on this particular slide, highlighting microbes that when, and the microbiota, that when it's dysbiotic, so a decreased diversity, an elimination of the good microbes versus the bad, you get disease. So in the distal ileum or the colon, necrotizing enterocolitis in the newborn, or inflammatory bowel disease. In the esophagus, the eosinophilic esophagitis. In the stomach, H. pylori, and in the jejunum, things like celiac disease. And there's some elegant work going on now in a number of centers, in particular at Mass General with Alessio Fasano at a celiac center, where they're actually looking at how to prevent celiac disease in genetically susceptible individuals by changing and manipulating the microbiome with both diet as well as pre and probiotics. This lovely paper published about 1.5 years ago by Dick Haller and his colleagues looks at the different compartments within the gastrointestinal tract and the specific genre of bacteria that are in those different compartments. This one schematic shows how the relationship between obligate anaerobes and facultative anaerobes, highlighted by the orange and blue respectively, when they have a dysbiotic state, can result in chronic disease. So dysbiosis in fecal microbiota can result in diseases like NEC, IBS, and IBD. And our goal is both with the diets that we have our patients on, as well as the use of things like pre and probiotics, is to get that decreased diversity and dysbiotic microbiota back to a healthy state. Now, I talked a little bit in the beginning of this presentation about the role of exposures and what happens to the microbiome. And one of the most seminal papers that looks specifically at C-sections, and this is not emergent C-section, but elective cesarean delivery, and the overall risk of childhood food allergy. To orient you, the y-axis on this slide corresponds to adjusted odds ratio. And for those of you who forgot your biostatistics in medical school, odds ratio means the likelihood of allergy, the thing that you're looking, the condition you're looking at, and the exposure, that being C-section, and the overall risk. If the odds ratio is 1, that means there's no risk. If the odds ratio is less than 1, that means the exposure actually protects the person from developing food allergy. And if the odds ratio is greater than 1, that means that whatever the exposure you're looking at confers risk. And in this particular slide, in this concept of adjusted odds ratio, if mom had a history, did not have a history of allergy, and baby had that was born vaginally, then there was an odds ratio of 1. That is no risk for that infant to develop food allergy. The bar in the middle, if you look at a mom who had a history of allergy and the baby is born vaginally, then just that familial predilection or genetic risk of allergy in the mom would cause a 2.5-fold more likelihood that that baby would have food allergy. And in this particular trial, which I thought was an elegant study design, they actually did food allergy challenge at 1 year to 2 years of age to egg. And so if a mom has a history of allergy and the baby's born vaginally, it's 2.5 times more likely that baby's going to develop egg allergy by 1 year of age. If you look at the bar on the far right, and I think this is the most telling thing in this particular study, if mom had a history of allergy and baby was born by C-section, there was an almost 8-fold increased risk of that child developing food allergies by the gold standard of diagnosis by age 1. And that's been seen now in a number of studies over the last 5 to 10 years. So this paper published in Journal of Allergy and Clinical Immunology in 2014, birth by C-section, early antibiotic use puts kids at risk for eosinophilic esophagitis. This subsequent paper published later in 2016, do C-sections alter a baby's microbiome by hurting later health? And a very elegant study published in Pediatrics in 2014 looked at 2 large Danish registries born between 1977 to 2012. And if you control for every exposure from birth on, it was C-section alone that showed an increased risk of juvenile inflammatory arthritis, asthma, and IBD in this patient population. And lastly, about 1.5 years ago, a lovely meta-analysis and systematic review shows that C-section alone can run the increased risk for children developing food allergies. And they even looked at term versus preterm babies in this particular study published in Journal of Allergy and Clinical Immunology. So hopefully you see how risky C-section is in simply conferring risk for development of autoimmune and allergic disease. And it explains why there are 2 multi-center trials now going on looking at administration of fecal flora and or vaginal flora or both in newborns born by C-section to see if they can decrease the risk of development of allergies and atopic manifestations. Now, what does dysbiosis often confer? Well, you can see this list of GI diseases that we all as pediatric gastroenterologists deal with, ranging from celiac disease to necrotizing enterocolitis, and a dysbiotic microbiota has been described as associated with that disease. And in particular, I'm going to focus on allergy, decreased counts of good bacteria, lactobacillus and bifidobacter, and increased counts of C. diff, as well as, and this is the take-home point, a decreased diversity of the gut microbiota was seen in kids and infants and adolescents with allergy. What about the esophagiome, if you will, or the esophageal microbiota? Well, we used to think that the esophagus was a tube that connected the mouth to the stomach, and we now know it's an immune organ, and it has its own microbiome. And in this particular elegant paper published in 2015, the esophageal microbiome in patients, children with EOE, was very distinct from those of non-EOE controls, in particular the type of bacteria that were seen. In this paper published just a month ago, February 2021, a wonderful article that looked at eosinophilic esophagitis and the microbiota. This particular paper showed that there was a trend towards decreased diversity, so lower microbial richness in children with EOE, and different types of microbes in this less diverse microbiota in children with EOE led to the disease. So this is where dietary approaches and or use of probiotics or both and prebiotics may actually be able to influence outcomes, both preventing and or as a therapeutic approach to EOE. Okay, so first section, we talked about the microbiome and its development. We talked about it actually beginning before birth. I described the concept of dysbiosis and then its relationship to different diseases. And now I'm going to talk about the epidemiology of autoimmune, in particular allergic disease, and why we're seeing a dramatic increase all around the world, even in countries where these conditions previously were very low in prevalence. If you look at North America, allergy epidemiology in particular, allergy affects as many as 50 million Americans, up to 30% in some populations. Allergies are the leading cause of chronic disease in the US, and in particular, food allergies are most common in infants and children. In the UK, again, daughter's living in London, so she sends me articles that come about the UK news. This is published in 2016. A recent meta-analysis looked at an increase by a third in hospitalizations due to allergies in the UK over the last 5 years. National Health and Nutrition Evaluation Survey, abbreviated NHANES, run by the CDC, the Centers for Disease Control, showed that in the last 15 years, atopic dermatitis or eczema has almost doubled. So the y-axis is prevalence. That's the frequency of the allergic manifestation. The x-axis is year cohorts, and you can see that skin allergy or atopic dermatitis has almost doubled. And what's important to take home from this is that in children under age 5, the most dramatic increase was seen, in particular, those living in the southern US. Well, if you think about that, over the last 15 years, the lower southeastern and southwestern United States, which were predominantly rural, predominantly farming, have become more urbanized. And so as we decrease the number of good exposures in this very young population, we're seeing a rise in allergy and inflammatory disease. This paper just published this year looks at prevalence in US and Canadian populations and demonstrates again an increase overall in allergies, in particular in the very young population. And the take-home points from this particular paper shows that overall IgE-mediated allergies on the rise. The prevalence seems to vary quite considerably. And some food allergies, and I'm going to talk about this in just a minute, may not be IgE mediated. Now, it's important to recognize, and there's a lot of interest in this, is that more people believe they were allergic to foods than is the case. So it's important to get at whether it's an actual report or true allergy. Now, as I mentioned, there are two primary mechanisms for food allergy. One is IgE and one is non-IgE. And it's those of us in pediatric gastroenterology who have to worry about these non-IgE mediated food allergies. IgE mediated allergies are the ones that have the diagnostic tests. So the blood tests that you do, the RAS test, the Immunocap, those have tests that you can do to look for IgE mediated allergy. So anaphylaxis, well-defined mechanisms, easier to diagnose. Non-IgE, delayed onset, often have sort of symptoms that may be due to a number of other conditions, and no validated tests. So now I'm going to focus predominantly on those non-IgE, and particularly allergy or atopic manifestations that affect the GI tract. There was a lovely lecture just a week ago given by Dr. Menard Ketcher from Colorado on EOE. I learned a tremendous amount from that presentation, and it's important to recognize that EOE is one of those non-IgE mediated food allergy manifestations that's on the rise. So over the next few slides, I'm going to go through what are adverse reactions to food proteins in infants, children, and adolescents. EOE is clearly on the rise. If you read the emergency medicine literature, EOE and meat impactions are the single fastest rising cause of meat impactions in 11 to 17-year-old children presenting to emergency rooms in the US. If you look at our center, GI Care for Kids, in 2012, we were following 590 patients with EOE. In 2019, 1,435 patients with EOE, and that is not represented by us doing more endoscopies or adding more people to our practice. It's a true increase in the frequency. If you look at this at a big, broader scale on this particular slide, where the y-axis looks at incidence, that's new cases per 10,000, the x-axis looks at year cohorts, you can see whether you're in Hamilton County, Ohio, or Calgary, Canada, or the Netherlands, clearly EOE is on the rise. And in this paper published in CGH in 2018, which looks at it around the world in elementary pharmacology and therapeutics, 29 studies looking at the prevalence and incidence of EOE. EOE was pooled prevalence, 34 cases per 100,000, higher actually in adults than in children. But what was significant was there was a sharp increase in the pediatric population, much higher than what was previously estimated. So in this particular meta-analysis published just a year and a half ago, clearly EOE is on the rise worldwide. Eosinophilic or non-IgE mediated allergy affects other parts of the gastrointestinal tract, and those are shown on this particular slide, ranging from EGID, eosinophilic gastroenteritis, to dietary protein enterocolitis, proctocolitis, and the extension of, if you will, cow's milk protein allergy. Another entity which we're really now just getting our hands around, and there's actually some criteria to define it, we couldn't spell it 10 years ago, is FPIES, food protein induced enterocolitis syndrome. And in this excellent review in Clinical Reviews of Allergy and Immunology published in 2019, about a year and a half ago, FPIES was characterized by vomiting an hour to 4 hours, and oftentimes within 24 hours of the ingested food, dairy, so cow's milk proteins above and beyond any other food. Protein was the single most common triggering foods, but there were a lot of other different types of foods that caused FPIES to occur. Heavy reliance on detailed history. There's no test for FPIES, and acute management is IV hydration, antiemetics, and IV corticosteroids. What's important to recognize is now, as we are understanding the epidemiology of FPIES better, there's an infant form. We now recognize a school-age form, and there's been even some recent reports looking at an adolescent and an adult form of FPIES. Again, something that is a marker, if you will, of the overall rise in atopy around the world. So why is this? Why are we seeing a dramatic increase in allergy and autoimmune diseases? And I think one of the best slides that depicts this is this particular slide here, with, if you look at the figure on the left, incidence of infectious disease, the dramatic drop very clearly due to vaccinations, and incidence of immune-mediated diseases, the specific T helper exaggerated response shown in blue, so increase in Crohn's, MS, and asthma over the last 50 years. And the hypothesis is that a decrease in infections, a decreased exposures, a decreased diversity in the human microbiome results in an increase in immune and inflammatory disorders. As an example, a study done, published 2015, 1,000 Swedish children, and they found that those parents and where parents were interviewed, they found that those children who washed the dishes by hand were significantly less likely to develop eczema and somewhat less likely to develop allergic asthma and hay fever than those that stuck the dishes in the dishwasher. So if you want a evidence-based reason to tell your kids to do chores, there it is in this slide. Or a friend of mine who is a health reporter for the New York Times, a geneticist says that any new parent should roll their child on the floor of a New York subway. And here's why. It's a paper published in Science, which gets at the observation that children who are born on farms or in rural communities are less likely to develop allergies and autoimmune disease than those that are born in the city. And in this particular paper, they actually demonstrated a mechanistic reason, and that is that farm dust actually induces appropriate diverse microflora and an immune response that protects children from asthma and allergy. Or if you think about this more philosophically, should you really pick your nose? Or maybe instead of that 5-second rule when you're eating at the dinner table, maybe that should be lengthened to 15 or 20 seconds. The bottom quote by Meg Lemon, a dermatologist who deals with a lot of atopic dermatitis, is involved in a number of clinical trials. She said, I tell people when they drop food on the floor, please pick it up and eat it. Now, I'm not making recommendations to our audience about that, but I think it raises some important provocative points about the fact that as our environment has become cleaner, our immune system has less exposures that are good and then is relatively unprepared, and thus you see disease develop. So now probiotics, prebiotics, and how can you affect the microbiome? And I'd like to think about managing our gut microflora like managing a nice, healthy, green, lush lawn, which everybody likes to have if they have a house with a lawn. And you've got two ways to actually enhance the richness of your lawn, your microbiota. Either feeding, done with prebiotics and diet that promote the growth of good microbes, or probiotics, or the ultimate probiotic, fecal transplant, that reseed the good microbes. So what are the definitions? A probiotic are live microorganisms that, when administered in adequate amounts, confer health benefit on the host. And I actually had the blessing of being on the FDA a panel that recognized the genus Lactobacillus and the genus Bifidobacter as generally recognized as safe for human consumption. A prebiotic is a substrate that is selectively utilized by the host microorganisms and enhances good bacteria, enriches the diversity, and results in a health benefit. A symbiotic is a mixture of both prebiotics and probiotics that confers health benefit on the host. And so diet is a key influencer of the microbiome, and whether you're breast or bottle fed clearly changes the type of microbiome that you have. And the gut microbes differ in studies that have been done in the last 20 years, either with clinical outcomes or microbial outcomes. The gut microbial composition differs between breast and formula-fed infants. Breastfed infants have higher abundance of Bifidobacter and Lactobacillus, whereas formula-fed infants, higher amounts of Bacteroides, Clostridia, and Enterobacteriaceae. This is not a new concept. Elie Metchnikoff, who passed in 1916, so well over 100 plus years ago, suggested that ingested bacteria could have a positive influence on microflora in the intestinal tract. And he hypothesized in his writings from the last century that lactobacilli were important for human health and longevity, and promoted yogurt and fermented foods. So even before we got into yogurt and FODMAP diets and gluten sensitivity, Dr. Metchnikoff was talking about the importance of these foods for health. Now, my son, who listens to me sometimes ad nauseum, practice my talks to him, said, hey, he looks like a former comedian. So maybe they were separated at birth. So Robin Williams and he actually look amazingly similar. Moving along. Lactobacilli have a number of different strains that have been looked at in a variety of studies. And I'm going to talk about the ones that have the most evidence as used for probiotics, as does Bifidobacter. And there are other microbes, in particular, you all may be aware of Florastor, where you have Saccharomyces boulardii, and that has been used in a number of clinical trials and didn't quite make GRAS status. If you think about this sort of as a schematic and ongoing circle, you have a tridimensional communication between people in the bottom part of the inner triangle, the colonizing microbes, and the probiotics, which for time's sake, I won't go through, but have a number of functions within the gastrointestinal tract. And I don't want to miss the point here to recognize that the products of the gut microbiota also have important roles. In particular, they're byproducts that even have neurochemical or opportunity to influence the enteric nervous system and actually affect emotional and psychological health. Prebiotics are food ingredients that benefit the host by stimulating good bacteria, typically non-digestible oligosaccharides, and there are a number of bacteria that generate energy from these fermentation products. In milk, it's those human milk oligosaccharides that are most important, in addition to the microbes that infants get in being breastfed that help with enriching the microflora. And there's been a number of studies that looked at prebiotics and as well as probiotics in changing outcomes. One of the first papers published at the Proceedings of the Nutrition Society about 5 years ago looked at altered gastrointestinal microbiota and irritable bowel syndrome, and now some tremendous data from Doug Droseman and his work at UNC Chapel Hill, or Bill Che and his work in University of Michigan about probiotics, prebiotics, and the low FODMAP diet at definitely influencing IBS outcomes. This is what the FODMAP diet is, so fermentable oligosaccharides, disaccharides, monosaccharides, and polyols, and they're different types of the FODMAP diet. But again, this utilizes or exploits prebiotics, and in some of these studies, they've actually looked at probiotics and changing the microflora to then result in normalized gut motility and decreased visceral hypersensitivity and good health outcomes. Even gut-brain, so the microbes and the brain interact both directly through interaction between the gut microbes and the enteric nervous system within the GI tract, or indirectly through microbial products and neurotransmitters that influence the brain. And so there's a tremendous amount of exciting research looking at the brain-gut connection and the role of microbes in potentially influencing outcomes. Breast milk human oligosaccharides can help balance the microbe system and in addition to enhancing the developing immune system, but also help the growth of normal pathogens. And there are even longitudinal studies or cross-sectional studies that have looked at functional disease and whether infants were breast or bottle fed, and the role of human milk, both the prebiotics and probiotics, in affecting outcomes. Obesity, another example of where the gut microbes, and I had to include in this talk this seminal paper published by Turnbaugh and his colleagues. So on the top, you have a low-fat diet. These skinny mice, if you will, have a lean microbiota. They're then given to a germ-free mouse living in a germ-free environment, and no matter what they eat, these mice stay skinny. On the bottom, you have a obese mouse that their poop is obese in terms of its microbial makeup, and you give that to a germ-free mice and they become obese. And you could actually then feed that now obese mouse in the lower right-hand corner the poop from the skinny mouse and actually make it skinny. So, and when you look at the calories consumed, these mice consume the same amount of calories. So take-home points, what you eat changes the makeup of your gut microbes. What you eat also change how your microbes function. And so this whole role of the metabolic syndrome and obesity in the microbiota is an incredibly important area for us to be focusing on in terms of helping result in healthy outcomes. And there's a number of studies, and these are shown, 3 of them on this particular slide. Fat flora, gut bacteria differ in obese kids, to obesity, weight gain message starts in the gut, to a Yale-led study that actually published about 4 years ago, which links gut bacteria and childhood obesity. Now, what about prematurity and being a setup for an altered microbiome and potential consequences? Clearly, C-section birth, less chance of being breastfed, the NICU microbes, which have been exposed to tons of antibiotics and lots of drug resistance, antibiotics, you have a delayed establishment of the microbiota, decreased diversity, and inadequate immune development, decreased gut barrier, and so it's a real setup for disease. Early recognition and aggressive treatment of NEC improves clinical outcomes. And NEC, as we all know, and I tease my neonatology colleagues, babies that had NEC are the gift that the neonatologists give to us pediatric gastroenterologists that keeps on giving. And this course of webinars had a wonderful lecture from Val Coran, who talked about small bowel and the intestinal rehabilitation areas where there's lots of exciting research. NEC is particularly significant in preterm infants. Now, why am I saying this with respect to probiotics? This is a Cochrane analysis published in 2014, 24 eligible trials included. And in this meta-analysis, enteral probiotic supplementation reduced the incidence, new cases of severe NEC and all-cause mortality. And there was no evidence in all of the studies reviewed of nosocomial sepsis or systemic infection. The conclusions of this Cochrane think tank was that enteral supplementation prevents severe NEC. I think it's important to have some caution with this. And Jay Kim, a neonatologist who's at Cincinnati Children's, who actually trained with me at Sick Kids in Toronto, I think his take-home point in this up-to-date review published 2018, we need to really establish a standard regimen that optimizes the type of probiotic that we use, whether it's single strain or multi-strain, and control measures that look at good outcomes. The AGA clinical practice guidelines just published this past year, actually key first guideline for preterm infants who are low birth weight, under 2,500 grams, specific probiotics can prevent mortality in NEC, reduce the number of days required to reach full feeds, and decrease the duration of hospitalization. So to bring this presentation to conclusion, what I hope I've done is show that probiotics aren't just replacing the bad bacteria or adding to the good bacteria. They do a number of things with respect to the enteric nervous system, to the gut immune response, to help in enriching the overall diversity of the microbes. And there are a number of clinical outcomes that I've looked at, ranging from acute diarrhea due to viral pathogens, to decreasing the incidence of antibiotic-associated diarrhea, to decreasing crying time and irritability in infant colic. Another cautionary note, and this elegant article in Clinical Gastroenterology, Claudio De Simone from Italy, who highlights the fact that this is an unregulated market really around the world. And probiotics commercially available have exploded. And so it's in terms of their availability. So it's important really to recognize there have not been any head-to-head clinical trials comparing one probiotic to another, and that the properties of probiotics are often strain specific and safety and efficacy related to that specific formulation and not to be generalized to other probiotics. The FDA has a nice a review published in 2018 on the specific microorganisms and microbial-derived ingredients that can be used, and they constantly add to this. So this is a good resource for those attending this presentation to look at. And in particular, two more recent provocative papers, one in IBD that looks at how dietary interventions to modulate the gut microbiome may become the way we have precision medicine in treating inflammatory bowel disease. And then in a paper published a year and a half ago in Nature Reviews, which looks at probiotics and prebiotics in intestinal health and disease, and actually which specific strains of probiotics have a long history and safe, effective use. Some of the up and coming ones like Akkermansia in particular, and the uses for these specific organisms. So where are microbiota? They're found throughout the GI tract. And remember at the beginning, I said the microbiome is the mixture of the microbiota, the microbes, plus their byproducts. Who are they? In early childhood, very stable. What we now know is that actually, ability to intervene may even be later childhood. In adults, Firmicutes and Bacteroides are the two predominant phyla, and there's a great deal of diversity. The microbiota regulate the immune system, maintain gut structure, and they can be manipulated by diet, such as prebiotics, probiotics, and combining them, symbiotics. I think one of the most elegant articles to leave you with about how taking an animal model of malnutrition and then expanding that to the clinical setting in countries where there's high rates of starvation, protein malnutrition, kwashiorkor, or overall malnutrition marasmus in these two particular articles. So the effects of microbiota-directed foods in nautobiotic animals and in undernourished children. So to summarize, in fact, these authors actually looked at the makeup of a malnourished microbiota. And then after the animals were nourished, the nourished microbiota, or after the nutritional rehabilitation occurred, and then targeted that type of microbiota in malnourished children. And that paper actually got published in the New England Journal just this past year, where they actually looked at how to change the microbes and, in fact, health of growth in stunted, undernourished children. So to bring this to conclusion, what I hope I've done is given you a real good overview of how the infant's immune system develops and interacts with the gut microflora. I've provided the epidemiology of why pediatric allergy is something in North America and Latin America and around the world we're going to be seeing more and more of, and some of the hypotheses as to why this might be existing, and the importance of the gastrointestinal microbiome in health and disease. And then reviewed for you how probiotics and prebiotics, and didn't talk too much about fecal transplant, the ultimate probiotic, can alter the microflora and change health outcomes. And for another resource, this is actually updated annually, a clinical guide to probiotic products available in the US. It comes just from a few months ago. And with that, I will conclude, and we'll open it up for questions. Thank you so much for your kind attention.
Muchísimas gracias. Ben, are you there with us?
I'm here.
Thank you. Thank you very much for that very nice presentation. Very, 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 very interesting presentation. Vamos a hablar un poco sobre el tema de microbioma intestinal y alergia. Y este es un tema muy interesante, sobre todo porque podemos tener sobre este tema diferentes perspectivas. Por ejemplo, Benjamin Gold vive y trabaja en Estados Unidos, es el presidente de NASPAGAN, una sociedad que reúne Canadá, Estados Unidos y México. Estos son países que muchos consideran países del primer mundo, que tienen obviamente un nivel de desarrollo importante, en donde obviamente, como él lo expresó en su conferencia, lo que es enfermedades autoinmunes, enfermedades inmunoalérgicas, enfermedades inflamatorias están en aumento. Sin embargo, yo, por ejemplo, soy el presidente de la Sociedad Latinoamericana, que reúne a 21 países, de los cuales 19 son considerados países en vías de desarrollo. En nuestros países todavía la prevalencia de enfermedades infecciosas es muy alta. Sin embargo, este mismo fenómeno de aumento en la prevalencia de enfermedades alérgicas, inmunológicas, inflamatorias se ha descrito y se está describiendo y lo estamos viendo en muchos países de nuestra región. Entonces, a pesar de que pertenecemos pareciera a mundos distintos, podríamos tener algo en común y tal vez sea estas alteraciones del equilibrio de la microbiota intestinal. But I'm talking about our different perspective over this tendency of a higher prevalence of immunoallergic, inflammatory, and autoimmune diseases. And I'm talking about different perspective because you are in North America, of course, you represent NASPGHAN, which includes Canada, United States, and Mexico. And I represent Latin America, which includes an important number of third world countries where the prevalence of autoimmune and inflammatory diseases is still relatively low. We still have a higher prevalence of infectious diseases, but the same phenomena is on the rise. It's been described over certain areas of the region. So maybe what we have in common is dysbiosis, the microbiota disbalance, and implications in all these immune inflammatory diseases. What do you think about this?
I think your point is very well taken. And in fact, if you look at data from around the world, in countries where there's lots of infectious exposures early on, in particular exposures such as parasites, which drive a Th2 type response, and children have a more robust immune system, if you will, you aren't seeing some of the things that I was describing, such as autoimmune and allergic disease. But just like the presentation last week from Dr. Menard Ketcher and some of the data that I presented to you now, EOE we're starting to see, which is a non-IgE mediated allergic and inflammatory disease, if you will, that we're starting to see in many countries that you weren't seeing this condition as well. And I think if you look in the future, the next 10 to 15 years, you're going to start to see some of these particular conditions. If you look at countries, for example, in Asia, where EOE was also very, very low, IBD is low, and allergic and atopic diseases in general, we're starting to see many more of these particular diseases. So I think what's happening is, again, an overall change both in diet, in infant exposures, increase in elective C-sections, that then decrease diversity in the microbiome and thereby predispose children, even who are not at risk by mom or dad and an inheritance for allergic disease. So I agree with you. It's definitely not in the frequency in Mexico and Central and South America, but these conditions are going to be are on the rise, and we're starting to see more. Hence, I think an opportunity to maybe even do some of the things that we are playing catch up, if you will, in terms of dietary approaches, in terms of recognizing who's at risk, and even potentially manipulating early on in life as far as protein choices and administration of probiotics to reduce the likelihood or risk of disease. Great question.
Very good. Vamos a hablar un poco sobre el tema de cómo intervenir. Sí sabemos que hay una relación entre disbiosis, sabemos que el microbioma intestinal del niño comienza a desarrollarse durante las etapas de la vida intrauterina y se desarrolla durante los primeros años de la vida, y sabemos que alteraciones en ese desarrollo del microbioma intestinal conllevan a disbiosis, y la disbiosis a su vez eleva el riesgo de desarrollo de toda una cantidad de patologías y enfermedades. Vamos a hablar un poco de intervenciones y el momento para hacerlas. We're talking about, there's a first question in the chat about this window of opportunity. We know you have showed us, we know the data that shows that the intestinal microbiome starts its development through intrauterine life, but it's very strong during the first years of life, of postnatal life. And of course, it's influenced by all these several factors you clearly explained. But let's talk about intervention. And there's a question about what would you consider the best moment for that creates a window of opportunity to positive modulate, to make a positive impact and try to induce a positive modulation of the infant microbiome?
So outstanding question. And I think as I pointed out to you in the presentation, it's that first 3 years of life that really is the ultimate window. And if you can't change how the baby's born, then early on in terms of selection of diet, if they can be breastfed, then breastfeed. If there's donor milk available, actually, you lose some of the opportunity for exposure to skin microbes, but you still get some of the other byproducts by giving donor milk. And even in breastfed babies, there are data to suggest that actually administration of pro and or prebiotics can enhance immune response. So say, for example, you've got a family where there may be a history in siblings, older siblings, or in parents and then in relatives of allergies or other types of autoimmune disease. That might be a population we're intervening even from early on, either in diet, so choosing the type of proteins that the baby's exposed to if they can't breastfeed, or if they can breastfeed, considering administration of probiotics. There have been at least a half a dozen papers, Reinaldo, that have actually demonstrated even giving probiotics in the third trimester of pregnancy might in fact actually influence and decrease allergic risk in an at-risk child. So there may be opportunities even in the third trimester of pregnancy, or even in breastfed babies, where you can intervene in that window being the first 6 months to 3 years of life. Protein choices, how early you introduce foods. If you look at the way the American Academy, and I think this has changed around the world, based on the EAT study that was published in the New England Journal of Medicine, which looked at peanut exposure, that in fact early exposures to food antigens potentially can enhance immune response and thereby decrease risk for overall allergy. So excellent question. And I think that's where more research, and particularly in populations where these type of diseases aren't as common, and we're starting to see an increase in looking at ways to influence diet, finding those right periods of time, that window of opportunity as you described, where you can intervene.
I think you mentioned the answer, because this question is a two-part question, and I think you mentioned something about it. La pregunta tiene una segunda parte que incluye el tema de intervenir sobre población sana de alto riesgo. Si existe una situación donde se justifique y existe una evidencia que soporte intervención temprana en población sana de alto riesgo. We're talking about, you mentioned this in your answer, but maybe you could elaborate a little bit more. Is there indications and formal evidence that supports the intervention of a healthy population, high-risk healthy population? Should we intervene? Should we work with healthy children of high-risk families?
So, there is data, and there's been really only one, and it needs to be duplicated in different populations. This was done in Europe, the Genie study that looked at initial randomization of at-risk babies from birth, over 2,000 infants, to 4 different formula types. So they were looking at protein choice, not necessarily at probiotics and prebiotics. The control group was intact cow's milk protein. They had an extensively hydrolyzed whey-based protein, an extensively hydrolyzed casein-based protein, and a partially hydrolyzed whey. They were given that, so you got enrolled in that by a flip of a coin. And for the first 4 months, and if there was breastfeeding, that was allowed too, which I think in some ways changes some of the outcomes. But nevertheless, 4 months on this protein choice, and then they followed those infants, not out a year, but 3 years, 6 years, and there's now just published a couple of years ago, 15-year data. And they looked at incidence, meaning new cases of allergic manifestations, either atopic dermatitis or eczema, allergic rhinitis, or asthma. And what they found was in the extensively hydrolyzed casein-based group, and to a lesser extent, but also in the partially hydrolyzed whey group, that even out to 15 years, just in that first 4 months of life, being given a different protein source, you actually decreased the overall incidence of allergic disease. So if you think about that, if I was going to redesign that study, I would have added a pure breastfeeding arm. So I would have one of those groups of infants completely and exclusively breastfed for at least the first 4 to 6 months. And I would have added a probiotic arm and then looked long-term. And there's data from Italy that have actually looked at that and demonstrated that in at-risk children, if you administer changing the protein source and adding a probiotic, you can actually decrease outcomes long term. So am I recommending it across the board? Not yet, but I do think that there's some very compelling information to your really good question that says that there might be in those at-risk patients, so strong family history of A to B type of allergy or an autoimmune disease, of potentially changing outcomes by dietary changes, trying to enhance everything you can to breastfeed, and if you can't, protein source and prebiotics. Great question.
Thank you very much. Okay, you just answered two questions that our colleague Jose Cadena from Mexico City dropped on the chat. And now we have another question from Jose Vera, who is in Bogota, who's trying to figure it out. This is a hypothesis, maybe. What do you think about probiotics as an option for the prevention of COVID-19? Maybe do you think there will be a place for probiotics in such a worldwide scenario?
So very, very good question. And intriguing because in fact, there's actually data, 3 papers published just this past month that look at dysbiosis in patients with COVID and predominantly gastrointestinal symptomatology. And there's actually some very intriguing data looking at the influence of the gut microflora actually decreasing or attenuating the response of COVID and not just GI response in patients who get infected. To Jose's really good question, there's actually data, at least in rotavirus vaccine, even in breastfed babies, that if you give probiotics, you augment the secretory IgA response and in fact actually can enhance the response to rotavirus. So although this study hasn't been done, and maybe Dr. Vera is a prophet, there may be an opportunity to potentially augment the vaccine response, the response that people get with COVID vaccine by enhancing the microflora and thereby enhancing the immune response. And at least it's been shown in rotavirus vaccine and now a number of studies that actually secretory IgA response is enhanced with probiotics. And those studies, Culturella or Lactobacillus rhamnosus was the probiotic that was used. So again, really nice, good questions. And I think things that really, I think, raise some important points that these are things that we may be doing more in the future.
Thank you. Okay, now we have another question. We leave Bogota, we go to Santiago de Chile, where Dr. Paul Harris is located. I'm going to read it for you. I'm going to translate it for you. It's written in Spanish. Everybody can read it. I mean, Dr. Harris says, bacteria have been forever in the digestive tube. And, you know, a long time ago, there were no C-sections, there were no antibiotics, and children used to die because of infections, you know?
Right.
So from an epidemiological point of view or perspective, is there data that shows that a reduction, is there a reduction in child mortality based on modification of microbiota? Is there data that show that you can, if you modulate microbiota, you can really translate that intervention into a reduction of infantile mortality?
Paul, Professor Harris, very wonderful question, and greetings, my fellow H. pylori aficionado. Actually, the best model is NEC, which I just gave you a sample of, which believe it or not, and there was only one pediatrician on the AGA panel that looked at their technical report, which just got published this past year. But if you look at necrotizing enterocolitis with the initial hypothesis that newborns that are born less than 30 weeks gestation are exposed, lack the exposures, so and have high risks for necrotizing enterocolitis. In that model, in a Cochrane analysis published in 2008, and the subsequent Cochrane analysis, which I presented to you in these slides in 2014, and in the AGA technical review, they actually demonstrated not just a decreased incidence in NEC, but also in all-cause mortality. So, has the study that Dr. Harris is elegantly describing been done yet? Not yet, but I do think that this gives you a model, if you will, that potentially could be extrapolated into other settings where, in fact, modulation of the gut microflora, decreasing in pathogenic bacterial colonization can actually result in decrease in all-cause mortality or death. So maybe there might be an opportunity, and again, the epidemiology really still needs to be done. But using that NEC model, it gets at, in a way, the question that Dr. Harris is elegantly asking.
Very good. Talking about NEC and the paper you mentioned at the end of your lecture, the AGA probiotics consensus, ¿no? And specifically in gastrointestinal disease. El doctor nombró al final de su presentación un trabajo, un paper publicado en Gastroenterology, que es un trabajo de posición de la Asociación Americana de Gastroenterología en relación a la evidencia que soporta la indicación y la prescripción de probióticos en patologías gastrointestinales específicas. Ellos revisan la evidencia basados en 8 patologías que van desde enfermedad diarreica aguda hasta enfermedad inflamatoria intestinal, pasan un poco por el tema de enterocolitis necrotizante y en cada indicación, en cada patología revisan la evidencia que existe en torno de si hay razones para prescribir o no probióticos en los diferentes escenarios clínicos. Y una de las partes en donde la evidencia parece apuntar o parece haber una evidencia bastante importante en torno a este punto es precisamente con el tema de enterocolitis necrotizante, prevención de enterocolitis necrotizante en el prematuro. In the paper you showed about the statement of AGI, one of the issues where the evidence is quite strong, quite impressive, is precisely the premature infant and the NEC prevention with different strains of probiotics used. It seems to be that there's a very good evidence that supports the use of this group of strains in the prevention of NEC. What do you think? Would you have experience using these groups of strains preventing or trying to prevent and reduce mortality in preterms from NEC?
So great question. Eager to answer that in a way, because one of the things that I think has been difficult, and that's why the neonatal NICHD neonatal network, which is a large consortium of neonatal centers funded by the NIH in the US and actually in Canada as well, wasn't ready to say that all newborns over 30 weeks gestation and over 1,500 grams need to be put on probiotics, because if you read all the studies, some of them had single strains, some of them had multiple strains. And your really good point in that technical report, they actually suggest multiple strains. And it makes at least biologic sense that at least multiple strains that have been tested, a VSL3 was what it used to be called. It's now Visbiome, which is a combination of lactobacillus and bifidobacter. And we've looked at that in Crohn's disease patients, but not necessarily in NEC. We've actually looked at that in infants who have short gut. So back to Dr. Corin's presentation when she was talking about intestinal rehabilitation, there is data to suggest that multiple strains may in fact enhance the ability for intestinal growth, intestinal rehabilitation, and getting them off of parenteral and onto enteral feeds. Again, head-to-head trials, to your really good question, still have not been done, but I think that those are the type of studies that are ongoing now, and we'll start looking at those questions.
Sí, la respuesta de Ben tiene que ver un poco, bueno, estamos haciendo comentarios sobre el trabajo o la publicación de la Asociación Americana de Gastroenterología. Y una de las cosas que uno puede concluir cuando revisa esa publicación es que toda la evidencia apunta hacia cepas específicas, es decir, cuando hablamos de probióticos pareciera que estamos hablando de algo genérico, pero cuando revisas la evidencia, existe cierta evidencia que podría justificar el uso de ciertas cepas específicas en ciertos y específicos escenarios clínicos. Eso queda muy claro en en la publicación. Cuando hablamos de enterocolitis necrotizante, pareciera que hay buena evidencia en torno al uso de multicepas, es decir, varias cepas de probióticos que pueden tener de manera sinérgica un efecto o pueden resultar en un efecto beneficioso para el paciente. Pero a lo largo de toda la publicación y cuando se revisa la evidencia en enfermedad diarreica aguda, en enfermedad inflamatoria intestinal, tanto rectocolitis ulcerosa como enfermedad de Crohn, en síndrome de intestino irritable, en enterocolitis necrotizante, en toda una cantidad de patologías, toda la evidencia está en cepas específicas. Es decir, no podemos hablar de probiótico como un término genérico, sino que tenemos que pensar en cepas específicas para indicaciones específicas. What I'm trying to do is some thoughts about the use of, because sometimes this is a question that always comes up when we talk about probiotics, you know, because it seems like we're talking about some generic thing that is miraculous and benefit for everything, and that works fine in every clinical scenario. And data that, like the one that is published, that has been published by the AGA, is very specific in terms of strain-specific effect under certain specific clinical scenarios.
And that, I mean, Reinaldo, perfecto. That's the point that I think people need to take home, is that not every probiotic is the same. And the problem right now with the literature, at least the way, there's really only a handful that have a very specific strain used for a very specific condition for a defined period of time, with now, thankfully, both microbial and clinical outcomes. But your point is very well taken. And I think before we say, okay, you eat a plate of food and just sprinkle some probiotics on it and it'll cure everything. No, the use needs to be, at least for the time being, in very specific conditions in which, with very specific probiotics that have been employed in those clinical trials. And it's really until we start doing more where we look at head-to-head studies and we look at it in disease versus non-disease host and really seeing if you can actually affect outcomes, that we'll start to being able to determine whether you can extrapolate that data to other people. So very good point. It isn't, one probiotic isn't the same and it has to be that used for that specific condition. So good point.
Very good. Well, Ben, listen, there are no more questions in the chat, but I want to hear from you, because you work for the FDA, you represent a huge scientific society, and I want to know your thoughts about our role, is trying to intervene our health policy. What should we be doing? Because knowing all this evidence, knowing all this knowledge that we have now regarding the effects of our own microbiome of nutrition, early nutrition, the mode of delivery, etc., the use of antibiotics in certain scenarios. Now that we have all of this knowledge, what should we do with it to make people healthier and to avoid and to prevent disease? What do you think we should be doing as scientific societies, and what should be our policy in terms of how could we work side by side with our governments to try to strategize and try to impact our health policy?
So number one, data speaks louder than words. So start collecting data on diseases, prevalence thereof, outcomes, dietary choices and sources, exposures in different populations. Second, education. Just what you're doing, like tonight, like in the previous three, education, not just about diseases, but the biology thereof, how they're caused, manage ways to treat them, and more importantly, if there are ways to potentially prevent them. So sort of thinking before the disease develops, if there's a way to actually prevent them. And we as pediatricians, first and foremost, because we're responsible for the lives of children, thinking about ways that, so better nutrition sources for parents, educating them, and with people who are in families where there's at risk. So again, education, getting good data, and then partnering, collaborations like NASPGHAN and LASPGHAN, where we could potentially use the synergy, if you will, of our organizations, and maybe partner with the general pediatricians within the different countries, like NASPGHAN partners, for example, with the American Academy of Pediatrics. So when it comes to health policy and you have multiple specialties, if you will, with similar points that then governing bodies are going to listen to that and then get into health policy. So good data, education in terms of at least what the data says to, and then partnering, collaborations like LASPAGAN and NASPAGAN, and then partnering with our sister societies, the general pediatricians, in order to impact more people. Excellent point. And more collaborations like we have.
Sí, estas reflexiones finales tienen que ver un poco con qué hacer con todo este conocimiento, porque obviamente ahí está la información sobre el tema de cómo se configura el microbioma intestinal del niño, inclusive desde la vía intrauterina, cómo impacta sobre ese desarrollo del microbioma intestinal del niño la nutrición materna, la vía del nacimiento, la exposición temprana a antibióticos, el riesgo de disbiosis, su asociación con tantas patologías. La pregunta es qué hacer con tanto conocimiento para de alguna forma generar políticas de salud que tiendan realmente a la prevención de todas estas patologías. Y un poco esas son las reflexiones que le está haciendo, que tienen que ver con, obviamente, el análisis de la evidencia, buscar la evidencia, conseguir los datos que permitan justificar acciones específicas para prevenir enfermedades específicas, el estudio, obviamente, el trabajo de colaboración intersocietario. Y todos somos parte de una comunidad científica, de una comunidad médica que puede regionalmente, de alguna forma, impactar en mejores políticas de salud que a la larga tiendan a la prevención de tal cantidad de enfermedades. Así que un poco esas son las reflexiones finales para esta noche. Agradecemos, por supuesto, como siempre, al doctor Benjamin Gold, que nos ha acompañado desde el principio como presidente de NASPAGAN y que obviamente ha sido uno de los grandes favorecedores de que este programa se desarrolle. Ben, thank you very much for your presence, for your participation tonight. It's a pleasure always to listen to your talks, to your way, to very expressive way to express the knowledge. I'll never forget that recommendation about letting your kid roll out in the subway floor. Very interesting recommendation. But thank you very much for your participation and for your work. I'll see you soon, and we'll see you always through virtually or face to face.
Daniel, buenas noches. Yes, buenas noches, everyone.
Buenas noches a todos y muchísimas gracias. Gracias, Carlos, por tu participación. Gracias a todos que nos acompañaron. Buenas noches y nos vemos el jueves. El jueves termina el programa de NASPAGAN Visita a LASPAGAN. Dentro de 2 días, 48 horas, vamos a tener un último webinar de este programa con la doctora Rachel Rosen, que se conectará desde Boston. Y va a tratar un tema muy interesante, que es el tema de la enfermedad por reflujo gastroesofágico, algunas manifestaciones atípicas. Así que el jueves cerramos el programa, comenzamos en Cincinnati con el doctor Becerra, luego estuvimos en Chicago, luego estuvimos en Colorado, hoy nos conectamos con el doctor Benjamin Gold desde Atlanta y cerramos el jueves en Boston. Así que hemos recorrido casi toda la geografía. We've been all through the whole
United States,
North American geography. Okay? Thank you very much. Buenas noches a todos y hasta luego.